Hello again, this is the first video of our second session. So my name is Adolfo Floresaife, and here you have my email in case you want to write me some questions or any comment about this video. In this session we're going to go a little bit deeper into the central dogma of the molecular biology, and in this diapo we have a nice example of how genetic works. So I thought it would be nice to put it here. First of all, let's remember the most relevant points in session 1. So the four kinds of molecules that are mainly involved in life are nucleic acids, for example DNA and RNA. We have also proteins, lipids and carbohydrates. So DNA is a polymer formed of nucleotides, which would be the monomers, each nucleotide is formed from a pentose, a phosphate, and a nitrogenous base. And the DNA itself has a form of a double helix in an antiparallel form. When I say antiparallel, I mean that when one string goes from 5' to 3', the other one goes from 3' to 5'. So we have this something that it's called anti-parallel. The four bases of DNA are adenine, thymine, cytosine, and guanine. That could be abbreviated as or using the first letter, like A, C, G, and T. They are paired, and A is paired with T always, and C is paired with a G. It is always written in the direction of 5' to 3'. When you see a fraction or a portion of DNA written in or anywhere, it has always to be written in 5' to 3'. In eukaryotes, DNA is organized in chromosomes, which has a structure, and that these chromosomes can leave some portions of DNA accessible to express a gene or to read something in the DNA, or it could be not accessible. So that's why we have chromosomes. And because DNA can be so large, so the chromosomes can give structure, so it can fit into a tiny space of the cell that is the nucleus. Two of the most important information parts in the DNA are the genes and the regulatory regions called promoters. If you remember, promoters are very close to the genes, and we have another kind of regulatory regions called enhancers or silencers that could be a bit further from the gene. Transcription is a process of creating an RNA molecule using the DNA as a template. So RNA uses adenine, guanine, cytosine, and uracil instead of thymine. That's important. RNA is single-stranded, and there are many types. One of the most relevant is the messenger RNA. That is the one that is transported outside of the nucleus to the ribosome, where it will be read to synthesize a protein. Proteins, if you can remember, they are amino acid chains that have a specific function, and they are the product of the translation process. This translation process is in the ribosome, and I think that's pretty much it. And the last point I want to summarize here is about evolution. If you remember, evolution is a process where random changes in DNA sequence can give to an organism an advantage, or could give rise to a diversification of organisms. So that's like a big picture of evolution. In this session number 2, we're going to, as I've told you before, we're going to go a little bit deeper on the dogma. We'll talk about DNA genetics and epigenetics, transcription and retrotranscription. We're going to talk a little bit more about RNAs, translation, and proteins, and genetic diseases. I pretty much suggest the reading of the second chapter of the handbook of Bioinformatics. This second chapter is called Information Processing at the Cellular Level Beyond the Dogma. Please do not forget to write down your questions as far as they come up to you. So at the end of the session, you can write me an email or post it on the Slack, and I will be glad to answer all of your questions.